Hi, I'm Anna Blakeney. I'm an assistant professor at the University of British Columbia in the Michael Smith Laboratories and the School of Biomedical Engineering. Thank you so much to the American Society for Gene and Cell Therapy for this inaugural Science Communication Award. I am here today to talk to you about my experience with science communication and RNA vaccines and therapies, or the RNAissance as I call it. To give you an outline of what I'll cover today, I'll first start talking about self-amplifying RNA for those of you who aren't aware of this technology. I'll also talk about our first-in-human trial of an siRNA vaccine against COVID-19 that I was a part of, as well as my social media work, specifically on TikTok, which I think is a great tool for education and awareness. To declare my conflicts of interest, I'm co-founder of a company called VaxEquity, which develops self-amplifying RNA vaccines. I also serve on the scientific advisory board for a company called GenVax Technologies and consult for Replicate Biosciences and the Cystic Fibrosis Foundation. So to give you an overview of what my lab at UBC works on, there's really three pillars. So the first is RNA molecular engineering. So how do we design better RNA that expresses for longer, as well as in specific cells that we're trying to target? How do we design better nanoparticle delivery systems that gets our RNA cargo to the cells and tissues where we desire to deliver it? As well as thinking about the RNA frontier. So how do we design other vaccines using RNA, such as for bacterial pathogens? We're very interested in understanding more about the interactions between RNA, nanoparticles, and the immune system, as well as using RNA to express monoclonal antibodies, which can be used to treat cancer or prevent disease. One of the priority areas in the field is minimizing the dose of RNA, which we have lots of data on this from all of the clinical trials that have been done. So I'm showing you here some data from Moderna's clinical trial. This was a phase 1 trial where they looked at 3 different doses of RNA, so 25, 100, and 250 micrograms. And their primary outcome for this study was looking at this frequency and severity of side effects, which are up here. So gray is mild, blue is moderate, and yellow is severe. And what I think is visibly evident from this data is that as you increase the dose of RNA, you also increase the frequency and severity of side effects. So really minimizing the dose of RNA is a priority in the field, especially when we're thinking about using mRNA for non-pandemic contexts, such as a seasonal influenza vaccine, as well as other applications of RNA, such as therapeutics, where these side effects may not be as acceptable to patients. So for those of you who haven't heard of self-amplifying RNA before, I'll talk about how it's different from a typical messenger RNA. So I have the structure of both molecules here at the top. Here I have a typical messenger RNA, which you'll notice has some typical components such as a 5' cap and a 3' poly A tail, as well as an untranslated region, or UTR, at both the 5' and 3' ends, as well as our gene of interest or antigen code in the case of a vaccine. You'll notice that self-amplifying RNA has a lot of the same components. So it has a 5' cap and 3' poly A tail, as well as UTRs at the 5' and 3' ends, and our gene of interest. The main difference is these 4 NSPs, or non-structural proteins. These are derived from a virus and include a replicase enzyme that is able to copy the RNA once it gets into a cell. So I have a very simplified schematic here of how this is different between mRNA and saRNA. So on the right, we have messenger RNA, and as many of you are probably familiar, once a copy of mRNA enters the cell, it engages with the ribosome and you get translation of the antigen. For saRNA, it starts out very similarly. So we have one copy of the RNA, it engages with the ribosome. You do get some translation of your antigen from this initial engagement, but more importantly, you also get translation of this replicase enzyme. So this enzyme then goes back up and interacts with this initial strand of RNA, and you get many copies of the RNA. Here I just showed 3, but there's actually thousands of copies of the RNA in the cell. And because of this, you get much more translation of your antigen, even though you're starting out with the same dose. So in addition to getting a higher amount of protein expression, you also get a longer duration of protein expression with saRNA compared to mRNA. So here I have some data from an in vivo preclinical study in mice where we used an exact same dose and formulation for mRNA and saRNA. So 1 microgram of RNA formulated in the exact same lipid nanoparticle formulation. The RNA was encoding luciferase, so on the y-axis we have the luminescence, which is proportional to the total amount of protein that we're seeing, and we tracked the amount of protein expression over the course of 30 days. So you can see here for the mRNA that we see a large amount of protein expression initially, which dwindles within 3 to 5 days, which is very typical for messenger RNA. Whereas for the saRNA, you can see it takes some time to start coming online, and the expression peaks somewhere between 7 and 10 days, depending on the design. But even 30 days later, we're still seeing protein expression from the saRNA. So while I was a postdoc at Imperial College London, I was working with a team on a self-amplifying RNA vaccine against COVID. So I want to share a little bit about this story and then tie in how important science communication is for all of these genetic medicines that we're working on. So as many of you are familiar, to make a new RNA vaccine against COVID, we take the spike antigen that's on the surface of the virus. We encode the pre-fusion stabilized spike antigen in our saRNA. We then encapsulated that in lipid nanoparticles that were developed in collaboration with Acuitas Therapeutics, which is right here in Vancouver, and then started our preclinical mouse models. One of the really amazing things about RNA technology that's really adds the technology, but I think is surprising to many people in the public, is just how quickly we can make these vaccines. So I want to walk you through how, even as an academic group, we were able to move these into clinical trials. So on the 10th of January, the circulating strain was sequenced and made publicly available. This was early in 2020. On the 20th of January, our team at Imperial accessed this sequence. I then flew to the campus here in Vancouver at UBC to interview for my now position at MSL on the 21st of January. So as you can imagine, this was an incredibly busy time. We ordered our DNA oligos to start our cloning on the 22nd of January. We assembled our genes into a plasmid on the 28th of January. We then confirmed that our saRNA vaccine was expressing the spike antigen as we had expected on the 12th of February, and started our immunogenicity studies on the 13th of February. We started our rat toxicology study on the 30th of March. We submitted our paper on this preclinical work on the 22nd of April, and then started our clinical trials on the 19th of June. So even as an academic group, we were able to move from designing a new RNA vaccine into clinical trials within 6 months. And larger companies such as Moderna did this reportedly even faster in just about 66 days. So this is an incredible technology, right? Obviously useful for outbreaks and pandemics, but many different applications moving forwards. I want to show you some of the data that we had from our preclinical studies. I won't get much into this because I really want to focus on the science communication today. But one of the things that we were really interested to see is just how low of the dosing that we could go to in our preclinical murine models. So I'll walk you through all the groups here. At the time, because we didn't have a positive RNA vaccine control, because none of them had been approved yet, we used electroporated DNA as our positive control, because we knew this typically worked very well to give both good antibody and cellular responses. We also had a negative control, so a rabies vaccine, to make sure our antibody responses were specific. And then we used 4 different doses of our saRNA formulated in lipid nanoparticles, so 10, 1, 0.1, and 0.01 micrograms. And then we compared this to the antibody levels that we observed in patients that had recovered from COVID-19. So on the y-axis, we have our spike-specific IgG as determined by ELISA, and these mice received injections at 0 and 4 weeks, and then we measured antibody levels at 2, 4, and 6 weeks. So you can see here from our electroporated DNA, we have a pretty good level of antibodies after the boost, 10 to the 5th nanograms per mil. But our saRNA was, even at the same dose, was over an order of magnitude higher at over 10 to the 6 nanograms per mil. We see a really nice dose response curve here, even down to our lowest dose of 0.01 micrograms, which was still giving us an antibody level of 10 to the 5th nanograms per mil, and remarkably was clearly much higher than the antibody levels that we were seeing in recovered COVID-19 patients. A lot has changed since this data was published, and obviously this is against the wild type strain, but we see how potent these saRNA vaccines are when formulated with lipid nanoparticles. So everybody always wants to know, where is the Imperial College vaccine now? So at the time that we finished our combined phase 1-2 clinical trial, there was already 2 vaccines in the UK that were approved, and so the team pivoted to targeting different mutations as well as booster doses. We started a company, as I mentioned before, called VaxEquity that is now working on developing this technology even further. And they recently announced a partnership with AstraZeneca to further commercialize this platform. So one of the things I wanted to point out is, we're not the only people working on this technology. There is actually now an approved saRNA vaccine that was developed by HDT Bio, which is out of Seattle, which was approved in India against COVID-19 at a dose of just 5 micrograms. So really starting to capitalize on using a lower dose using saRNA compared to mRNA. But what I really want to think about, and I think the importance of communicating about this science really comes from thinking about where this technology is going. So we've seen that it works really well for RNA vaccines, for viral pathogens, but I wanted to walk you through too. This is not a slide I prepared, but I actually got this from BioNTech's website, but I think it does a good job of summing up where all the applications that we're using this technology for. So obviously they say today we have the approved mRNA vaccine that was developed in collaboration with Pfizer and BioNTech. Tomorrow, so this is things that are already in the pipeline, so including mRNA vaccines against other infectious diseases, mRNA cancer vaccines, CAR T-cell amplifying mRNA vaccines, as well as systemic immunotherapies. But beyond that, thinking about different applications such as autoimmune diseases, allergy, inflammation, regenerative medicine, and other therapeutics. So the potential of this technology is huge and obviously going to make a big difference. I think we've really entered this era of mRNA medicines. One of the things I realized during the pandemic, and really what's motivated me to do more science communication, is just realizing that if nobody wants to take the vaccine, there's no point in making it. So I'm showing you a few maps here on the right, which was a paper published by Heidi Larson in 2020, which looks at kind of 3 different attitudes towards vaccines. So at the top, we have vaccines are safe. We have in the middle, vaccines are important. And at the bottom, vaccines are effective. And you see there's this gradient of respondents where blue indicates that respondents strongly agree, whereas orange indicates that respondents do not agree. And you'll see that there's quite a disparity between, lots of people think that vaccines are important, it's majority blue, but there seems to be a misstep kind of between, or a disconnect between thinking vaccines are safe and vaccines are effective, even though we have all of this clinical trial data that points to the fact that mRNA vaccines are both safe and effective, right? So I'm really interested in kind of understanding, where does this vaccine hesitancy stem from? It's not new, but I think was, very much in the spotlight during the pandemic. And what is the missing link between believing vaccines are important, which, the majority of people do, but not safe or effective? And I think this really comes down to science communication and our responsibility as scientists. So why is public engagement important? To me, I think this is really motivated because the pace of science is just not matched by the pace of public communication. So as of January 2020, there were a lot of scientists that didn't know what an RNA vaccine was, but now billions of people have gotten them. So of course there is just a lot of questions that people have, and it really moved quite quickly during the pandemic, as obviously it needed to. But I think it left a lot of questions brewing in the general public that may not have heard anything about RNA vaccines before they had the option to get one. So one of the initial efforts that I was a part of was for kind of trying to get more information out there, was doing Reddit Ask Me Anything. So if you're not familiar, Reddit is another social media site, and they have a whole page called AMA, or Ask Me Anything, where they bring in experts. And you can see here, this was doing it with my colleague Paul at the time. And you announce the event, and then people put in their questions and you answer them for a certain time period live. So it was really great. This was organized and supported by the Imperial Communications team, so it was great to have help for that. We found that it was actually a really great way to interact and answer questions, and it kind of enabled us to see what people were asking questions about, but definitely difficult to manage all of the questions that come in without help. So as we were doing this, you can see on the left here that it got lots of upvotes. It actually made it to the front page of Reddit, and so got a lot of attention because of that. And very coincidentally, the next day I received a call on my office phone, and it was an organization saying that they were Team Halo, which is an organization that was started between the United Nations and the Vaccine Confident Project. And, the pandemic was a weird time, but I didn't even know my office phone number. And so when people called it, I always thought it was a little bit sketchy, but I agreed to take a meeting with them the next day. And they told me their whole goal was to connect scientists and clinicians who were working on COVID-19 to the general public to improve vaccine literacy and confidence, specifically over TikTok. So I had never been on TikTok before, but I was very interested to see, is this a good social media platform for being able to interface with the general public and educate people about RNA vaccines. So this is exactly what I did. Team Halo has grown a lot, and their overall goal, I think, was a really interesting approach. So people were obviously using TikTok a lot during the pandemic, and they targeted many different countries worldwide. So they have a 66 guide in over 20 countries worldwide. So this is not just in the UK or in Canada where I am now. And they have, the last time I checked, at least over 95 million views across social media. So obviously been very impactful way to actually interact with people and get the word out and kind of really embrace this social media for scientific purposes. So when I first went into it, I kind of thought TikTok was just a platform for dancing teenagers, but I really started seeing beyond that. So obviously I started in October as a part of Team Halo. I had never done it before, so it took a little bit of getting used to, like any new social media platform. But you can see now my channel has grown, so I have over 250,000 followers now, which is a really cool opportunity. What I found is that it's quite an elegant platform to show people exactly how we make and test vaccines and directly confront misinformation. One of the reasons I think TikTok works specifically is that it's video, so they can actually see you, and I think there's a level of sincerity involved in it. But the videos are also short and engaging and offer a palatable amount of information. So unlike this, a lecture where I can really dive deep into a subject, I really have to think about when I'm making a TikTok video, what are the main takeaways that I can communicate in 15 or 30 seconds so that people are really able to learn in this time. So this has been highlighted in many different media sources, which I never would have expected at the beginning, but I was featured on BBC News a few times. I also ended up being covered by Vogue UK, which was very unexpected, but cool to see scientists in a fashion magazine, as well as other magazines such as Bustle, which is also did a story about the scientists using TikTok to fight COVID misinformation. So one of the really cool opportunities I had as part of using TikTok to kind of educate people more about RNA vaccines is that I have an opportunity to really recognize the science between the COVID-19 vaccines. So I was very lucky to have kind of two superstars in the field. So on the left is Bob Langer, who is a professor at MIT, who is also a co-founder of Moderna. And on the right, I have Peter Cullis, who is a professor here at UBC, who founded Acuitas Therapeutics, which is the company that formulated the Pfizer and BioNTech vaccine in lipid nanoparticles. And I've just had really amazing responses to the videos, especially where I have other scientists involved. And so you can see on the right here, there's some comments like, thank you for saving the world, real life superhero. I would love to get it. Thank you for saving lives. Thanks for all your hard work. So grateful to you, Peter. And it made me really realize that people appreciate scientists, but they also have no idea who they are. These people are very well known to people in the field, but definitely not to the general public. And so I think this is a great opportunity to make more scientists household names. And I think this really helps to kind of tie in all the work that has been done in these fields, for decades now. So I wanted to highlight too some of kind of the TikTok comments and outcomes. So one of the unexpected outcomes for me was actually being able to have meaningful conversations and comments from people. So like one of the users said, getting my second dose on Thursday. Someone asked, I just received my first dose of Pfizer, just curious, will I have protection after the first dose? Already eager to get the second one. Someone on the left, which says, don't trust it yet. And I said, I see. Are you just waiting for more time to pass? And you can see a lot of people responded to this. So yes, same. And so it really is a way to open up conversation around this. And I try to do this in a non-judgmental way so that I can actually kind of understand where people are coming from as well. And I've gotten a lot of feedback like this as well, just saying I have both of mine, but thank you for opening up a judgment-free space to talk about this. So unexpectedly, it's not all just trolls and dance videos, but it is actually a place where I think we can really genuinely communicate about our science and scientific progress. So the impact to date, there are now over 100 expert guides that are involved in Team Halo. There are over 4,000 videos that have been made as a part of this and over 340,000 comments. We've gotten over 240 million organic views, so quite a number of people have viewed these videos, and even more views supported by a partnership with Facebook. And the team has also been featured in over 100 media articles, including Vogue, the BBC, CNN, CBS Global, and Times of India. So in conclusion, kind of my main takeaways from this is I think there really is a gap, and this is not just for mRNA vaccines, but for lots of genetic medicines and all different fields as well, between the scientific progress and what the public is aware of. And I think closing this gap is really important and will also help to enable acceptance of these new technologies. I think each engagement strategy, such as Reddit or TikTok, it falls on a spectrum of quality versus quantity, and so kind of have to decide what you're going for, but they all have their pros and cons. But I also think social media can be a useful tool to educate and get people more interested in science, and so I'm always trying to encourage more scientists to go on TikTok. So thanks very much for listening. I would like to acknowledge all of my current and previous lab members, as well as our funding sources. And with that, I will take any questions. Thanks so much for listening today. I really appreciate this opportunity, and please feel free to reach out if you have any questions in the future.